something interesting, biofilm. People talk about biofilms in bacteria, which is something similar to this. And we will talk about also the role of this biofilm infection, which is also connected to resistance of bacteria and infection. And I think now we are reaching so, Erik, please.
Thank you to again organizers for inviting me. I'm a little bit on the outskirts, I think, in terms of what I'm doing at this conference. So to illustrate maybe first why I feel a little bit out of my depth here, mainly actually presenting, I think, methodology. I can tell you that what we typically been working on is colloidal science. This is my main expertise, and we've been working a lot on biomedical particles, trigger drug delivery systems, in protein interactions with polymer nanostructures and so on. Also taking that now to bioplastics and nanocomposites. But another big part that we've been doing in my group for ages is that we try to develop new methodology from physical chemistry and apply it to sort of measure colloidal properties in biological systems. And that's sort of where today's lecture fits in. And I'm also not basing it on a decade of experience on what I'm talking about. I'm actually talking about the last 2 years of research, or more or less 1 PhD student and 1 postdoc, or 2 PhD students and 1 postdoc. And it's sort of motivation from medicine will be something like this. Now we have a disease called cystic fibrosis. I think most people have heard of it, which is not very, very common disease, but it's quite excruciating. And we don't really have a good cure for it yet, just like cancer. And what it does is that it mutates a gene. So some people carry this mutation, and this gene is sort of controlling the hydration of the mucus that you have in your lungs. So if this is not pumping out the chloride ions as it should do, then your mucus becomes very dry, and it's very difficult to get it out of your lungs. So in principle, you're suffocating yourself by having this problem. That part is possible to medicate to some extent, but the real problem comes from the Pseudomonas aeruginosa, bacterium, an opportunistic pathogen, is very good at colonizing this state and creating another mucoid biofilm in this structure. And we basically have no way of exterminating this type of bacterial infection. So our interest was a little bit on trying to understand from materials perspective, how come that the bacteria, which usually responds decently to antibiotics and can be eradicated, in this state, it basically cannot. And this is, of course, is a research topic that many others also have attacked. And the sort of consensus in the field is that this has something to do with a specific mucoid biofilm structure. So what is that? A biofilm is a community of bacteria, for those who have not encountered them before. And an interesting sort of fact about this, which is coming on the next slide, is that the major part of the biofilm community is the matrix of polymers that they are creating around themselves. And this matrix of polymers in the mucoid biofilms is a bit special compared to other Pseudomonas biofilms, in that is extremely rich in alginate, which has some of the properties that Grady was showing before of creating, in the presence of calcium, very, very say tight gel, extremely hydrated structures that are also anionic. And these type of biofilms also include a lot of DNA, which also is highly anionic. And many of the antibiotics that are used for treated these type of diseases is cationic. And so the main idea has been that the tight mesh of hydrogel that they create, this is highly anionic, can then sequester or stop or maybe completely block the diffusion of the antibiotics that you try to add. And that was sort of the idea that we started with in this project, and also thinking about adjuvants on how to change this structure. But I will not really talk about that because it turned out the hypothesis from our perspective is kind of wrong. Another thing about biofilms is that there are communities that have larger structures on a micron scale, on a 100 micron scale, and so on. And if you look at it in any textbook, it's very well described what a biofilm looks like and how it develops over time. And it's more or less derived from 2 species, Pseudomonas and E. coli, and they often depict a scenario like this, that you grow into large tower-like structures, and from these you release planktonic bacteria that create new biofilms downstream somewhere. And that is sort of the main picture that you have. But if you just look at the mucoid Pseudomonas and the standard Pseudomonas from which this picture is derived, the mucoid Pseudomonas actually has a completely different structure and is not very well characterized. And I think this is true for many types of biofilms. We have not introduced the structure very much here. So our sort of questions from this, coming from working a lot with hydrogels and so on, is that what if these structures have much more meaning than just how you release bacteria? Maybe they also control things about how antibiotics get in, how resistance develops and so on. And in that case, does it come from structure or from composition that changes through the structure? And yeah, again, so about in a biofilm, actually, the bacteria has synthesized most of the metabolic, the mass that they create to make this extracellular matrix, which is actually in contrast to us, which is producing most of the biomass as cells. So obviously, they're not doing this for altruistic purposes to polymers. It must have a reason. Another thing that sort of inspired me to show that I come to this topic is that when you look at especially biology and chemistry, it's not so common in physics, is that it's this very, very strong paradigm of that structure yields functions. You study structure, you study function at the same time. And we do that sort of on the macro scale, and we do it on the molecular scale. In engineering, we often do it to reverse engineer what function should be. But if you look at biofilm and microbiology research, it's very, very focused on omics, especially genomics, to try to understand how these communities work, which kind of gives you an average soup kind of picture. But I think it kind of lacks this detailed resolution that we might need to understand how they organize and talk to each other. So we also wanted to see if we could use some of our techniques to sort of fix this gap, which I think might have a function. This is why I'm giving this kind of methodology talk here. So the two main things today is the first part, which is published, where we studied whether the matrix can be reverse engineered to control or sort of elucidate what antibiotics actually do in order, or how the biofilm actually reduce the efficacy of antibiotics in the biofilm state. And the other one is again similar to what Grady was showing. So like going in and looking at the organization of the cells and organization of cellular matrix, which was sort of a goal after getting a strange answer to the first question here, and then finding out a little bit what we can do with our new techniques to learn about biofilm structure. So the first part then is about antibiotic tolerance. And the aim here was to take the hypothesis as it is, that it's the alginate structure. It's a special type of alginate you find in biofilms, which is not the same as people usually do in research because you cannot buy it. And whether DNA is, which is also often used, sort of combated in order to deliver antibiotics more efficiently in patients, whether this actually plays a role, and one should focus on the DNA by adding DNases and so on to these cocktails or not. And we also want to do this by seeing the microcolony formation that you often see in electron microscopy images of mucoid biofilms, if this is also part of the structural defense that the microbes have. So we're physicists, physical chemists, and also some of us are engineers. So we try to let's try to build this system from scratch. We know exactly what is there and see if the common sort of components play a role. So we developed bioink type of approach where we could print biofilms, where we sort of choose the composition of alginate, DNA, and seeded bacteria into this of another strain, which is not a mucoid strain, to see if it was affected by the properties of the gel. For this, we created increasing complexity. So we started with a non-acetylated alginate, the one that you buy from Sigma or whatever, and people typically use for research, which is coming from algae, obviously. And then we chemically modified this to correspond to the type of acetylated alginate that you find in biofilms with the same acetylation ratio. And then we also mixed in DNA into the bioinks and then printed these structures. We also wanted to compare that then to the real mucoid strain. So we made similar to the printed films. We also then, of course, cultivated the mucoid biofilms too. You'll see results from this too. So how does it work? You can either print these as beads, so we can print them as films in discs, depending on what kind of readout format we want to have. And what you get then is that if you seed them by controlling that the cells really are colloidally stable here, you can see them, and then they create these microcolonies on their own. That was our hypothesis, and it turned out to be right as well. You see on the next slide. And then when we have these type of artificial biofilms, then of course we can study whether we get the structure that we want, and then whether the antibiotic interaction with ECM is what we think. So first of all, we want to see if we get the microcolonies. This one can do with confocal microscopy. You will get images like this, more or less. With cryo-SEM, we could also then confirm that the microcolony sort of remodel only the immediate environment and leave the rest of the alginate matrix also that can be filled with DNA as it was. So what did we learn from then exposing these type of printed biofilms to different types of antibiotics? We used a large battery, so the main 6 ones are shown here that are often used in treatment of Pseudomonas aeruginosa. And this is just a control. And if we then would print them into a non-acetylated alginate matrix, then one would see that they actually already retain some resistance to the antibiotic. So here they've been exposed to 2 times the concentration of the antibiotic that would kill them or inhibit the growth, actually. So you should not see anything here if it was working. So with gentamicin, for example, and ciprofloxacin, there the matrix did not provide any protection, but for the other ones it did. If we then added this acetylation, that was one of the tests we wanted to have, whether this mattered or not. It seemed like the resistance or the tolerance, for antibiotics was moderately increased. And in the type of antibiotics which still were killing the bacteria, then it was a dramatic change actually in adding the acetylation. We're not really 100% sure why that was. If we then added DNA, which was then supposed to further block the access of antibiotics and increase the tolerance again, we actually saw the opposite effect, which we correlated a little bit with having studied the rheology and the mesh sizes and diffusions and so on of these matrices and with physical chemistry methods before, that it reduces actually the integrity a little bit of the biofilm, probably this 1 over this cationic effect of sequestering antibiotics. Another way that you can do this test is that you then study what is the concentration now that I need in order to still inhibit the biofilm growth. So instead of using the dosage that you would use for killing planktonic bacteria, you actually try to find what is stopping them from growing when they are in biofilms. And now the biofilms are again the printed ones and so on, and we cannot see the same effect. So acetylation mostly increases tolerance. When we add DNA, the tolerance go down. But I think you should also look at the numbers here on this type of logarithmic scale. And you see that the doses that you need are just tremendous. This is the reason why we cannot eradicate these biofilms, because this type of amount of antibiotics are extremely difficult then to deliver. So now the question is still, we have a higher tolerance, but is this due to that antibiotics actually don't get through the ECM, the extracellular matrix, as was postulated? We had a bit of trouble to develop a good assay for this and turn more in the microbiological direction, but still printing the different types of ECMs separately into small discs, and then sort of looking at the diffusion of antibiotics added to the Petri dish here, to the agar gel there. We then put our biofilm model in between, and then we collected an agar disc on the other side, and then used that one to look at the zone of inhibition to sort of a cannery in the coal mine type of bacterium that is very responsive then to this type of antibiotics. And from this, looking at whether the antibiotic actually diffused through here, the type of biofilms that we have, versus a control, of course. What we found out was a bit surprising to us. This is not what we expected. We saw that more or less none of these biofilm models that we, from physical-chemical perspective, had the same properties as the biofilms themselves. These are separate papers that were published before. Basically, none of them were significantly obstructing the diffusion of the antibiotics. It means that the bacteria are exposed to the antibiotics just like before, just like they had been in the planktonic state. There are very, very tiny differences there. So we checked, okay, is that because we have the wrong models? So we checked it with taking a mucoid biofilm instead that we've grown in this type of discs, and then we would have to have an extra protection layer disc here to not have a contamination with bacteria from the Pseudomonas bacteria before we go to the test organism, and then comparing it to control. Then we see that in most cases, it's still so that the antibiotics goes through very quickly with small differences to our biofilm models. But there are some cases like topramycin and gentamicin and colistin that you have on the top row here, There you have extremely retarded diffusion, or we actually don't see them diffuse through at all. The interesting thing here is that this is not all cationic effects. It's also part of some sort of steric obstruction of the larger type antibiotics as well. Anyway, that means that the sort of composition of these matrices cannot completely, like the main components, alginate and DNA, cannot completely explain the higher tolerance the bacteria are showing. If there's a barrier effect that we see in some cases, then the barrier effect must come from not the dominant type of polymers and anionic polymers that we see in the matrix. And the main candidates that are left are then two other big glycopolymers, PEL and PSL. But we still see this tolerance increase massively, so it must be from some sort of other encapsulation effect that then stimulates the bacteria into have a higher tolerance, really. Probably, I think actually they change the cell coating more because they're not going into the dormant state and so on in these young biofilms, which would be another explanation in much older biofilms. Yeah, since we're a little bit out of time, I then instead go on to the next questions that we had then. So if it's not really the interaction with the matrix from the antibiotics that matters, maybe it's the structure instead was what we were thinking about. And also we had started to suspect that the microcolonies that we would see in all these images from SEM and stained samples and so on in the literature, that maybe they were not really microcolonies after all, based on other research that we were doing. So we wanted to investigate that as well. So we turned into a project, we wanted to image instead the structure of Pseudomonas biofilms and then compare this standard strain where all the research has been done, which is called PA01, to the mucoid one and see if we saw differences between the two as well. So in our group, we work a lot with real-time measurements to develop techniques for doing very, very fast holography, which makes it possible to measure 3D motion of bacteria, especially close to surfaces, but also in matrices. And then we'll also look at surfaces directly with 2D type of fast microscopy. But what I'm going to show you now are instead using standard confocal microscopy, but in a more quantitative way than I think most people do for biofilms. And then also cryo-SEM, as you saw from Grady, because there we can get the ultrastructure, what the ECM looks like, and also confirm whether the fitting that we have to do to make statistical confocal microscopy, whether this follows sort of what we would see in the SEM, where we had much less statistics, but much higher resolution. So I told you we already suspected that a lot of the things that you see in the literature when you look at especially scanning electron micrographs on biofilms and bacteria are artifacts. And I think this is a good illustration of that, at least for these type of biofilms and bacteria, this is the case. So most literature actually just dries stuff down in order to use high resolution SEM in vacuum to look at the shape of bacteria and so on. But you actually change already the morphology of the cell itself when you're doing this, because this is very, very drastic. Most of the energetics that goes into keeping biological structures is usually from the entropy of water. I think most of us know that from proteins, roughly 50% of the energy that goes into creating the protein structure comes from the entropy of water. Now imagine what that does to structures that are usually much more hydrated, like the glycocalyx, the extracellular matrix, and so on. It's really take away the water, you take away everything, the structure, so you also then cannot do this structure-function analogy. You can also find paper where they're a bit more careful, do critical point drying, which is like taking away the worst, let's say, kinetic effects from drying, but doesn't change that you change the energetics. And in those cases, it's often where you see, this is actually a picture where it's not so strongly aggregated, but you often see clusters of cells like this, and you see a very meshy type of extracellular matrix in the background with little detail as you can trust. And in here, you see this type of cryo-SEM images where you will see many more of these then later. We see that the cells are actually quite alone here, and you can also start to see some structure inside the ECM here. So it really matters how you prepare the samples, and it's complicated because in the end, this is a vacuum technique, right? So you have to image in vacuum at the end and still keep the water there, therefore the cryo. It can be even more clearly, maybe when you look at films that were then prepared and then cross-sectioned, so we sort of break the sample, you can look at them from the side, And this is the same biofilm, but this one was dried. And you just see bacteria here creating an extremely dense mesh of bacteria, and there's basically no detail in the rest of the structure. While you keep the cryo-SEM, it's a very, very thick film, and you see dispersed bacteria into something which is a hydrated, mainly polysaccharide type of matrix. So water energetics here can really not be forgotten if you want to study structure. So the other method, I said, so probably many of you know how microscopy works, and confocal microscopy is sort of the standard microscopy with the highest resolution, but it's still limited by the point spread function, which is an order of hundreds of nanometers, and bacteria are about a micron big. So you have quite poor resolution compared to the objects you want to study, and especially if the bacteria then are reasonably densely packed or close to each other, you get a lot of overlap in the fluorescence signal, and it can be difficult to sort of filter this out. But since computers get a lot faster, you can be a sloppier and sloppier programmer and still sort of make a code that makes this happen. And you would have a lot of slices like this, that you take a slice at certain distances throughout Z space, and then you, of course, in an XY space from this, and you can have different types of dyes, and then create intensity maps of those that you then can start to treat with all the tools that we have from image analysis. So usually you have to go through steps from removing a lot of noise in the background. You have to morphologically close these images because of the fluctuations in intensity that you have. Then you have to make sure that you know where one bacteria starts and one or stops and another one starts and so on. And then what we used was that we also had knowledge from the SEM about the shapes and shape distribution of the bacteria. So we could fit them to something that was a simpler geometrical object, which greatly helps the statistics then later. And from doing all of this type of image analysis on processing the images in 3D, then you can get center coordinates, you get sort of information on the morphology. In this case, using a fitting object, you get a 3x3 rotation matrix that tells you how this is oriented in 3D space, which is then also related, of course, directly to the angles of the object. And with all of this, you can do fast calculations. So you can have tens and many tens of thousands of these bacteria and still sort of create a larger volume space where you know how they're oriented, how they are labeled, and sort of how they're organized in clusters. And also, if you look at this image, it looks like it's quite dense, but it's kind of an optical illusion because the type of density that you get here and the kind of thicknesses that you can measure for the films and so on, they agree very well, actually, with the SEM images that we use as a control. And now we can start to compare that. And what are the structures from, for example, then the mucoid biofilm with the PA1 biofilms, and we only study them now in the early stages where they grow for a couple of days before they form these mushroom structures. And our idea is that this is additionally interesting because at that stage, no one really thinks that they have an interesting structure, or it's actually, well, maybe a lot of people think, but they don't discuss it very much. And that's usually the case for the biofilms that you try to treat biomedically. You don't wait 5 weeks under perfect nutrition conditions and then try to treat. More often than not, you're in the earlier stage of biofilm formation. We do this. So we got good agreement for these basic measurement criteria for the films, but we can also then do a quantitative analysis, section this into different zones, and find out that the mucoid Pseudomonas have a higher concentration of bacteria or close to the surface than they have in the top layer, and the same thing is true for the PA1, but the difference is bigger for the mucoid PA. Another thing that we found out, which was surprising, because if you look in the literature as well, you expect the PA1 to be a much denser in terms of cell density biofilm than the mucoid PA. What we found is that giving them the same conditions and then like growth times and so on, so they grow for 4 days, what happened was that the mucoid PA is a much thicker biofilm, but it has more or less the same cell density as the PA1 has. And that did not really fit with our expectations. And it more or less means that for our conditions here, the mucoid PA must be dividing much faster than growing the biofilms faster. It's doing everything faster because it's also creating much more ECM than the PA1 is doing. So this is just hardcore working bacteria. What we also found was first, so you can also do this with SEM. Then you get statistics from the 2D instead. So kind of slightly worse statistics, but you can look at also how close are the bacteria to each other by calculating radial distribution functions. These things are very well known then from colloidal science to try to characterize self-assembly and so on. But I think this image is more interesting, which is showing you that when you look at these images, you don't really see any organization, any orders. You have to do statistical analysis to see what is there. And what we see is that there is some sort of clustering over around 8 to 10 microns and so on, where the bacteria are forming slightly correlated or higher density structures. That is what this type of Ripley function calculations are showing, which is also something used in material science. And this is true for both of the species. And what we also find is that the clustering is actually higher in the top layer, where you had a lower density of bacteria, than in the bottom layer, where you had a higher density of bacteria. But an important point to make is that the sort of values that we get for the Ripley function here shows that these are not microcolonies. These are well-dispersed bacteria that are just sort of still related to each other statistically in the position that they have. And of course, we get the same thing then from the cryo-SEM, but with much worse statistics. So we don't really have the same confidence as we would have for the confocal measurements. We look at tens of thousands of bacteria. Another thing that I also haven't figured out yet is that we can then look at sort of order parameters for this pneumatic order parameter, how they are aligned to each other, the bacteria as well. And then we find out that the ordering in terms of orientation is quite weak in most of the biofilm. But close to the bottom, you reach quite high pneumatic order parameters, which means that, let's say, I don't remember the direction, but say maybe most of them are standing like this, close to each other, and another point they're standing like this, close to each other. And this distance over which they are correlating is also quite long. It's on the order of 5, 6 microns or so on. So many cell distances away. They're still knowing what they are doing. And this is a little bit then puzzling because it means, sorry, I should go back one step, because thematic ordering of bacterial biofilms is discussed in the soft matter literature. But there is always found as a sort of response to crowding of the bacteria, being close to each other, because then you always get into the nematic phase for these type of objects, but we're not there. We're at a much lower clustering density. So somehow over a distance, they are still organizing. And I don't think that this is an artifact from cell division, because I think we sort of see, let's say, the clustering of the bacteria in the top layer might be due to that we're closer to the cell division event and the daughter cells have not migrated away so far. But in the bottom layer, this shouldn't be the case. So it seems like they can transmit forces over longer distances inside the biofilm matrix here. So to sort of finish off here, another thing that we found and was also a little bit unexpected is that in most biofilms, we don't really see any structure. It's kind of meshy so far. We haven't looked at so many yet. But in a mucoid biofilm, they should have this alginate dominated extracellular matrix, which we know has no structure because they looked at those to death when we were printing them. It does have that in the mucoid biofilm. We see this type of fibrous structures there as well. They don't have the same dimensions and organizations as the Gradimer images, but otherwise they sort of indicate again that this is some sort of glycoconjugate type of structure with a massive protein core and side chains. And we don't see them in the sort of top layer of the biofilm, and we don't see them in the PA01 biofilm either. So they seem to be unique here, and they might actually explain some of the deviations that we had between alginate biofilm done in the lab with the same mesh structure as the real biofilm, and the mechanical properties, which always seems to be slightly different, and also sort of follow shear planes and so on for the real biofilms, but not them for the printed biofilms. So I'm kind of out of time. I sort of summarized already the first part of the lecture about antibiotics. So just emphasizing the second part here, the new methods that we have of doing statistical image analysis of cryo-SEM and confocal microscopy can highlight such things as clustering, distance communications, different distance in organization, and things like this, which I think are very important when we try to understand antibiotics tolerance, but also this type of communication that you find in biofilms, because they also organize themselves at the end in quite complex structures where they spend an enormous amount of energy, metabolic energy, on creating a community, actually more than our cells would do, I would argue. So one should not sort of, like Michaela was asking about the spaces before, I think we should focus a little bit more on the space in between the cells, to put it that way, than the cells themselves and what they do, because there's a lot of physics, chemistry, and so on going outside. And if we don't know the structure, how this is organized, we will not learn very much, because those components don't tell the entire story. So with that, I should also thank people. So more or less all the work here was done by Goodness, also with her twin partner, Stefan Schandl, who was doing the synthesis of the polymers and the printing. And then Guru was mentioned before, so he was also supporting and training Goodness on the cryo-SEM. I also like to thank Alex, who is the bioink or the bioprinting professor at another university in Vienna, who helped us with this. And especially thank also to Olivier, who got me interested in this in the first place, and then left science for joining the evil empire, that is the university bureaucracy. But it means I have a friend there that can protect me a little bit as well. So it's all good. With that, I hope there's not at least one question I can have.
Thank you. Any questions, please? Yes.
What was the reason based on which you expected the diffusion of the antibiotic to be affected by the field?
Because it's all over the literature. That's basically the standard dogma, is this. That it either has to do so, there's different reasons why it might be hindered. It might be that, for example, mesh size is so dense that it takes a long time for larger molecules to diffuse through. But mesh sizes have been known for quite a long time for virological studies and so on. But then it's also the interaction directly with the type of molecular sort of groups that you find in these biofilms, where it's been then highlighted that the cationic antibiotics that are mostly used, they find a very polyanionic environment in there. And then I'm not sure why people think that this will completely block diffusion through, but that is the standard hypothesis of how this occurs. And well, it's more than a hypothesis because it's used also in how you create the treatments of cystic fibrosis. So in that sense, it's going beyond. I would say it's a hypothesis that in our hands is not true. But you add, for example, enzymes to cut up the DNA in order to deliver antibiotics so DNA is more efficiently. You add also kind of disruptors for the cationic structure in order to get the cationic.
Your cells, your whatever, bacteria, what is there, seems to grow just fine within this field, okay? Which means that the nutrients can diffuse as they like to that mesh. Okay? I would be surprised if the antibiotic molecule is much larger than the nutrients. So if the nutrient can just go as it likes, okay? The antibiotic also can go as it likes. So if there is anything in the field, must have something to do with a chemical reaction or something, molecular attaches to whatever causes the field. But this is not a problem of diffusion, it is just a problem of I get stuck. If I stick myself to the wall,
I would say that your intuition is mostly right, but of course there are also, previous to this work, they've also shown that, for example, especially tobramycin and gentamicin can be significantly hindered by putting a hypogeologist like this. Also that the hydrophobic interactions, some of these antibiotics are not that small, I mean they are bigger than most of the nutrients and the bacteria grow well, and there's been then a speculation about the stress. I think that might not be completely wrong, but I think it's more located to the different type of matrix environment that the cells have immediately around them, or I think that they have immediately around them, because this is where you have these other polysaccharide coatings like the PSL and PEL, they are enriched there. But again, in the literature, they only been investigated as extracted and thought as distributed. When we try to do the same, we realize that they are in this basically like a cell protein. Yes, please.
Yes, please. Thank you very much for the video. You mentioned that essentially you use more imaging techniques. So I remember the video and mobile, digital. Digital, you mentioned the future, the 3D, and the
So I can hit those.
about the digital, you mentioned the two futures, three-dimensional analysis, the two-dimensional analysis, and the laser-free capability. What do you think is the most important for radiography? I mean, I'm not a biologist, I don't know. Is it very difficult to stain bacteria or when they are identified with structures, or is it important, most important to add to the organization of the structure?
I think it really depends on the question. So from my perspective, it's very dangerous as soon as you start to label things and saying things. You always change it. What you're doing is that you use the physical interaction in order to see something, right? And you also don't know for sure whether the interaction partner is the right one. Photography specifically, there as well, you're diffraction limited, but the limit is slightly worse. You also cannot have a strong scattering background, so it's not so very useful when the biofilm start to grow, because then we lose a lot of the single information. On the other hand, it's extremely information rich. When you can focus the image, you lost almost all the information. That's what you do when you focus. In holography, we're taking the total face intensity information from the entire object, which makes it possible to use for identification without labels. This is other things that we do in this phenomenon. But we're using it especially to understand how bacteria interact with the surface to begin with, how different colloidal objects interact, because you can measure so fast. I can measure it as fast as my camera goes and get all the information from this. So that's where the strengths are there. But again, I have no biochemical information because I'm not staining, I cannot look in complex biofilms and get information out and so on. So it depends on your question, I would say. That's your question. So cryo for higher resolution nano, confocal for standard sort of size stuff, holography for if you want to have kinetics.
Any other questions, please? Yes.
I know myself. Yeah. So I will use it myself, and whatever technique you usually find something that's difficult or complicated. For example, one reason why we're not using super resolution microscopy instead of confocal microscopy would help is that you have to take real care about the heating of your sample, which is often a problem. There's no problem. I should talk to you.
Any other questions, please? Or we? Yes.
Yeah, so this is, for example, this next question, because I think understanding CM is important to understand how the phages in the end, right? So you ask about which components we find, or mainly see that. Yeah, so this again depends on the biofilms. We compare two different ones in this talk, and one of them, the absolutely main component is bacterial alginate, which is almost all of it. And the second biggest component there is DNA. If you take the standard PA1 Pseudomonas, it's the same species, right? It's a different strain. They don't produce alginate at all. So the main component is then completely different. Sorry.
Yeah, so there are alginases, for example, if you want to fix this one. And this has been added as well, but not super successful in the medical situation. autophage, generally everything that shows up polysaccharides is good, right? Because they are everywhere in every biofilm. The major structural component will always have polysaccharides that is really structured together. Now the things we can do, and often this is done, is that because the polysaccharide created, divalent cations is usually the glue that keeps us together, similar to what Daniel showed. So adding EDA and things like this also loosens up the structure. It almost never dissolves anything perfectly, right? But you can then measure the rheological properties change, the mesh size changes and so on, whereas in the phage is big, so presently it's then really well enough.
And just to add up for them, depending on sometimes the kinetics of the EDA or EGA to chelate in such a gel is very low. It should take days. We did even experiments with that. So you need a long treatment if you think to do the experiment, just to keep it in mind. Any other questions?
I think what you was thinking about the goal is the end point is similar memories, right? There's kinetics in between. But even with the similar memories, there's it's a big difference in the two states. So you have to have a high off state in order to replace.
Yeah.
I mean, if this is not present, then it doesn't happen all the time.
Yeah, exactly. Any other question? We discuss also later in a later time. Anyway, so now Erik, thanks a lot. Fantastic, really